So, question 5. Eukaryotic cells and prokaryotic cells have similarities and differences in their ultrastructure. Compare and contrast the ultrastructures of eukaryotic cells and prokaryotic cells. Firstly, ultrastructure just means what's inside the cell. So basically, that's your organelles. Right, so we'll start off with the similarities. It's just something you're going to have to just memorize, and there's 3 similarities. So they both contain a cell membrane, they both contain cytoplasm, and they both contain a ribosome. Now, differences. Here, the ribosome is also used as a difference because the size is different. So in eukaryotes, the ribosome has a size of 80S because it's composed of subunits 50S and 30S. Whereas in prokaryotes, the ribosome is smaller, being 70S, and that's because it has a subunit of 70S and 20S. Right. Another difference is that eukaryotes contain membrane-bound organelles, but prokaryotes do not. So, this includes your chloroplasts, your mitochondria, and your nucleus. But we don't really say the nucleus in here because we use the nucleus as a whole different example as another difference. So, if we move on, so if we say prokaryotes, no membrane-bound organelles, the next difference is that eukaryotes have a nucleus, whereas prokaryotes do not. They have a nucleoid, which is just a circular piece of DNA, which just unbound. Finally, is that prokaryotes have a cell wall made of peptidoglycan, or murin, whereas eukaryotes, not all of them have a cell wall. So we know that animal cells don't have a cell wall. Plant cells do, but here it's made out of cellulose. Fungi have a cell wall, but they're made out of chitin. And that's it. So, moving on. Some antibiotics inhibit RNA synthesis and protein synthesis in cells. Actinomycin D, rifamycin, and alpha-amanitin are antibiotics that work by binding to molecules in a cell. This inhibits protein synthesis. The scientists who developed these antibiotics had to find out which types of cell was affected and which molecule they were binding to. The table shows the types of cell that these antibiotics affect and the molecule that they bind to. Describe the structure of a DNA molecule. Well, firstly, just to go through it about why they do this, it's because when we discover new antibiotics, we want to see that if we give them to a human or an animal, is that gonna affect the human and the human body in a really bad, and is it going to cause bad reactions? So we do these to say, is it going to affect eukaryotic cells? Because if it does, they're not going to give it because it's going to be toxic to the individual. So we just want it to be toxic to the prokaryotic cells. So that's just some background knowledge about why they do this. But moving on. Describe the structure of DNA molecule. We know this is how DNA looks like. And this is your nucleotide bases. So first thing, what we say is that DNA is made of nucleotides. So we've got your A to T and C to G. And these are your nucleotides. Second thing we describe it is that the nucleotides themselves are responsible for making the backbone because they have a pentose sugar, which here is going to be deoxyribose, and that's your base. It can be an A or a T or a C or a G. And then you've got the phosphate sugar. That's one nucleotide. And many of these, if you repeat it, this is what will happen. That right there is a phosphodiester bond, and after you have so many of these, that's what's creating this backbone. And so what you say is that it's held together by phosphodiester bonds. Then, between the nucleotide bases, so A to T and C to G, we have hydrogen bonds. So between the complementary bases. Finally, we have two strands. So you have one strand here and one strand here. So you have two strands that end up making a double helix, which is this structure, because the hydrogen bonds are between the complementary bases, and so that twists the shape, and so it makes a double helix. Explain the effect of actinomycin D on protein synthesis. Okay, so actinomycin affects both prokaryotic and eukaryotic cells and affects them through DNA. And we know that it's talking about protein synthesis. Protein synthesis is made out of transcription and translation. And transcription occurs in the nucleus, the DNA, and translation occurs in the ribosome. And since this is talking about DNA, we know that they're talking about the transcription bit of protein synthesis. And transcription occurs because of the DNA unwinding. And so if the DNA is the one that's targeted, then if what's happening is that the DNA strands are not separating. They don't separate. And because they don't separate, you don't have the template. And so, without the template, you have no transcription, because the antisense is missing. The antisense strand is not available, sorry. That's the strand that acts as a template, the antisense strand. And so no transcription means no mRNA produced. Now, final mark is that because it affects the DNA, and DNA, the code, the genetic code is universal. And so that means that these bases, the A, T, C, G, are found in every living cell. And so prokaryotes, we know that also go by these bases. And so that means that the DNA is affected in both types. And so, explain the effect of rifamycin and alpha-amanitin on protein synthesis. Okay, so rifamycin only affects prokaryotics, and alpha-amanitin only affects eukaryotes. Okay, but both work because it inhibits RNA polymerase. So, we know that RNA polymerase is involved in transcription, because that's the enzyme that's going to be, if this is our strand, the antisense strand, and we're going to be forming here the mRNA, this enzyme, the RNA polymerase, is responsible for creating the mRNA strand. And so if the RNA polymerase is affected, that means transcription is inhibited. And so you have no mRNA strand. Right. Why? Because let's explain it. Because the RNA polymerase it's a dog barking. Because the RNA polymerase is inhibited. Because dog. So the transcription is inhibited because we said that the antibiotic is going to bind to the RNA polymerase, and that's an enzyme. So the enzyme is inhibited, and so the transcription is inhibited. it seems obvious that you don't have to state it, but you do. There's the mark, because you already read in the question that said RNA polymerase is the one that's going to be affected, and so it seems obvious not to include it, but you have to include it, because the best way to go about answering these questions is always to have it in your mind that the examiner knows nothing. So you cannot assume that something is already present and they must know it, or it's in the question. You always have to include it in your answer and act as if they know nothing. So always explain everything. Alright. The final mark is that, okay, they both contain the RNA polymerase, but how come rifamycin only affects prokaryotics and alpha-amanitin only affects eukaryotic cells when they both have the same enzyme? Well, that basically means that the RNA polymerase, the enzyme, does not have the same structure in these cells. So the RNA polymerase in eukaryotics is different to the RNA polymerase in eukaryotics in terms of structure. They both may have the same role but different structures, and so these two antibiotics are also different in structure. And so that antibiotics, the rifamycin, will affect this structure of the RNA polymerase, and alpha-amanitin will affect this RNA polymerase.